Okay, so welcome everyone. It's my distinct pleasure to invite Professor Paige Novak. She is the Joseph T. and Rose S. Ling Chair in Environmental Engineering and the Department of Civil, Environmental and Geoengineering at the University of Minnesota in the Twin Cities. And Paige is originally did her bachelor's at the University of Virginia in chemical engineering, and then both a master's and PhD in civil and environmental engineering at the University of Iowa. And she has many awards. She's a fellow of the Association of Environmental Engineering and Science Professors. She was editor-in-chief of Environmental Science, Water Research and Technology. So many other awards as well. But on top of that, Paige is just a really incredible person. I mean, she has mentored a lot of students and really helps people in their careers, including myself. So it's a great pleasure now to actually have her here to speak for us today, at least remotely, and we hope that she will be able to visit us in person at EAWAG in the coming years. So with that, please help me in welcoming Paige.
Thank you, Serena. I really appreciate it. I'm going to shrink everyone's faces down, so if you cringe, it's okay. I won't notice it. But I really appreciate the invitation. I'm really happy to be here remotely. I'd love to be there in person. But I'll be talking today about some work that we've been doing over maybe the past 7 years or so on encapsulated microorganisms and using these microorganisms in this encapsulant for enhanced wastewater treatment. And I met with Serena and Dave Johnson before we started, and there were some interesting conversations that we had about spatial organization that I think got me thinking, and so I'll touch on that as I go. But I wanted to just start by kind of laying out the main motivation for some of my work, a lot of my work, and probably a lot of your work as well, and that's the mitigation of climate change. And so this is a quote from a paper that came out in 2014, a while ago. And it says that the global carbon footprint should be reduced by 60% between 2010 and 2050 to achieve the climate target of a maximum 2 degrees centigrade of global warming. We're not doing very well on this so far. And so we really need to think about all the different sectors, transportation, environmental engineering, being able to kind of address climate change so that we can start to move closer to this goal. And so that's where my work in wastewater comes in. And when you look at the statistics around wastewater, in the US alone, we have about 85 billion cubic meters of per year of wastewater that's generated. And this includes both industrial wastewater and municipal wastewater. And its treatment accounts for about 2% of the total US electricity use. And so this is a huge amount of electricity use that is attributed to wastewater treatment. Globally, I found this statistic from the UN. It's estimated that about 330 billion cubic meters per year of municipal wastewater is generated. And this would be the wastewater that is being treated. And this wouldn't include industrial wastewater. And so there's a huge need to think about treating wastewater in a way that doesn't contribute to climate change and actually can help mitigate that. And so thinking about how most wastewater is treated, and this is a US-centric view, I'd be curious to hear if it's the same in Switzerland. But what we tend to do is we have a lot of mid-sized and small industries, especially food and beverage industries, that discharge their wastewater, mix it with the municipal wastewater, and this is all blended in a single wastewater treatment plant that then typically uses aerobic treatment to degrade the waste materials, and then it's discharged to a receiving body. If you look at that industrial wastewater, especially again thinking about food and beverage wastewater, most of these waste streams are pathogen free. They separate the sort of the toilet wastewater from the industrial wastewater. And some of these consist of wastes that could be treated anaerobically for resource recovery. A lot of these food and beverage industries have really strong wastewater in the tens of thousands to hundreds of thousands of milligrams per liter of BOD or COD. And so these are really high strength wastewaters. And you could treat those to recover energy. The other thing that happens in the US anyway is that these industries will typically pay the municipality to discharge this wastewater to the sewer to have it blended and then treated. And so this is a cost to the industries, plus they're not getting any benefit from this wastewater. If you look at the municipal side of this, these waste streams are dilute, they contain pathogens, they contain personal care products and hormones, and they also contain nutrients. And when you think about nitrogen removal in particular, it requires aerobic treatment, at least for ammonia oxidation. It requires low carbon concentrations and long biomass residence times. And when these streams are blended, it makes resource recovery much harder because now we've diluted that industrial wastewater. And it also can make nutrient treatment harder because now you've added strength to that municipal wastewater. And it also makes the treatment more energy intensive because the amount of energy you expend is typically proportional to the amount of COD or carbon that's present. And so if we think back to how we can change the system, how we can improve it to mitigate climate change, we need to start thinking about what we can do as environmental engineers to help get to this climate mitigation. And so some of the things that come up are we can separate industrial and municipal treatment. We can, break those two streams apart. We can develop lower energy, low operation and maintenance, small footprint, anaerobic industrial wastewater treatment options. And the reason why there are so many descriptors here is a lot of times there are small industries like a dairy or a brewery, a candy manufacturer, and they don't have a lot of space. Their goal is not to treat wastewater. Their goal is to make dairy products or beer or something like that. And so they really need a low O&M system that's small footprint in order to treat their wastewater if they're going to do it in-house. The second thing we need is low energy nutrient removing municipal wastewater treatment options that facilitate long residence times of the biomass. And so when we thought about this problem, what we came to is the idea of encapsulation, a biomass being one promising solution for these issues. And we can use this similar or the same technology basically to solve both of these problems. And this shows images from our lab of encapsulated biomass. These are alginate encapsulants. We've used a bunch of other types of encapsulants that I'll talk about through my talk. But if we zoom in on these encapsulant beads, what we've got is these alginate beads are this matrix, this mesh that are trapping organisms inside. So these little beads are filled with bacteria. And what we can use these for is the retention of specific organisms that we encapsulate in this matrix. And we can design the matrix to some extent. There are definitely bounds on this to achieve certain internal environments in this encapsulant. And so when we look at this, the encapsulant is typically made of cross-linked polymers or polymerized monomers. The compounds that we've worked the most with are alginate, PVA, and PEG, and we've also used combinations of these compounds. These encapsulants can be chemically modified. We can use different crosslinkers. We can cross-link them in different ways, and we can actually use mixtures of polymers. And we've done a lot of work recently with alginate-PVA combinations. And we can also coat these encapsulants to achieve different outcomes. And then this encapsulant surrounds or traps the organisms inside. And we can control the physical nature of these beads to some extent also make them in different sizes. And with the alginate in particular, we're really good at controlling the size. We can go anywhere from about 1 to 1.5 millimeters up to about half a centimeter. With some of the other ones, PEG, they tend to be a little larger. And I'll talk about that as I go. But this is basically what we're talking about. We're talking about this matrix that then holds those bacteria in place. So when we look at encapsulation, there are a lot of similarities to biofilm systems. And this is kind of a nice way of thinking about this because you can imagine some of the same benefits that you get from a biofilm system. We do get biomass retention, and you can separate the hydraulic retention time and the solids retention time really easily. We can also use the encapsulation system to develop microenvironments or niches within this encapsulant bead, like you would with a biofilm system, where you see diffusion of oxygen coming in from outside the biofilm, and you can get different niches inside that biofilm. We can get the same sorts of things happening in an encapsulant bead. There are some unique advantages to encapsulation, and that's that we can control the initial inoculum. I'll talk a little bit more about that later. And that can allow for a rapid startup. And you can also, to some extent, tailor that encapsulant to achieve certain outcomes. And so if we look at this retention of specific organisms, this should give us some specific benefits to our overall goals. It should enable long residence times for these small footprint anaerobic treatment systems. And some of the systems that we're kind of comparing or benchmarking our encapsulation system against are things like anaerobic membrane bioreactors, where you have a really energy intensive process to achieve the same thing. To achieve this separation between hydraulic residence time and solids residence time, you have to push that water through this membrane that fouls, and you can end up with high energy costs actually using that system. The other alternative that has been around for a while are more large footprint biomass retention systems, which doesn't necessarily work for these mid-size industries. It should also enable effective nitrogen removal by retaining certain slow-growing organisms. And with this, we're interested in both Anammox organisms, anaerobic ammonia oxidizers, and aerobic ammonia oxidizers, both of which tend to grow slowly. With respect to this protection and control of the internal environment, some of the benefits that come from that are that we should be able to protect the anaerobic organisms that are present from toxins. And this should facilitate both simultaneous aerobic and anaerobic processes within the encapsulant and potential control of substrate concentration. In order to make this actually work, we don't see a lot of encapsulation systems out there in the world. There are some, but what we need is a better understanding of the interactions between the biomass that's encapsulated, the bulk environment out here, and then the encapsulant itself for effective performance and also predictable performance. So where we focused in our lab is in two areas. We've looked at total nitrogen removal and municipal wastewater treatment. And this shows some of our lab scale reactors there. And down here, you can see these little encapsulant beads. And we've also looked at COD removal and methane production in high strength industrial wastewater. And we've done a lot of work with a brewery partner here in town. And here you can see again some of these little encapsulant beads in about a 3.5 liter reactor. And here's an image of some of these beads. We've added powdered activated carbon to these beads to help stabilize the bead. And I just want to show a couple of types of the experiments we've run, just so that as I'm going through my results, I kind of mix all of this together. You can get a sense of what I'm talking about. We've done a lot of mass transfer experiments, and this shows our diffusion cell system, where we would make kind of a little puck of a particular encapsulant, and then we can study the diffusion of compounds across that little puck of encapsulant. And we can get then the diffusivities of these materials. We can also do partitioning experiments, and we've done a bunch of those. We can do small scale batch experiments, and we've done those as well. And then we've got small flow through experiments that we do, lab scale experiments. And this shows, again, the nitrogen removal system. And down here, I've got, probably making you sick by now, an image of what some of our systems look like for the high strength wastewater. And then another type of experiment we did is this horribly messy looking thing. And this is actually deployed at the brewery, where what we've run is these larger flow through experiments that are more automated and controlled. And what we've done is we've actually run a side-by-side anaerobic membrane bioreactor so that we can compare the performance and compare what's going on in our encapsulated system with this ANMBR, which is sort of the state of the art. And I'll talk a little bit about that data as I go as well. So again, some of the things I'm going to zoom in on are this environment encapsulant interaction. And with that, what we're really thinking about is how we can tailor the encapsulant to control or understand the mass transfer in the system. With respect to the biomass encapsulant interactions, I'll talk about that as well. What we're focused on there is trying to control the escape of organisms out of the encapsulant, understand the migration within that encapsulant. Can we get migration, or do we get differential growth within that encapsulant? And then also, do we get growth? How do the organisms grow and adapt within that encapsulant environment? And then finally, I'll show some data that we've done, or we've gotten on trying to develop models to predict this performance with time, so that we can then use this to start to design systems that perform better. So I'm going to start with the environment encapsulant interaction. And with this, the experiments I'm going to show, what we were looking at is measuring the diffusion and partitioning coefficients of substrate, products, inhibitors, and trying to understand how that differed within these different encapsulants, and using that to choose the encapsulant. And again, we did a lot of diffusion experiments with these encapsulate pucks, and then we've done a lot of partitioning experiments. And we've looked at the effect of material, crosslinking, and the coating in these systems. So the data I'm going to talk about is, I've just zoomed in on partitioning coefficients. And what I show here are some of the experiments, some of the results that we've gotten for alginate only, silica gel coated alginate. So this is a way of stabilizing that alginate material, but it also adds a different type of interaction, chemical interaction there. And then we've also done a little bit of work with polyethylene glycol. We're doing a little bit more of that now, PEG. And then you can see all these different chemical species that we've looked at. Oxygen, this is taken from the literature. All the other data we've generated, but the oxygen is taken from the literature. And so what we've done is we've measured these partitioning coefficients in these different materials. And I just want to highlight a couple of these so you can kind of see what we can do with this type of data. When you look, when you look at the nitrite data and the wastewater organics data, a couple of things that I want to point out is when you look at different materials, alginate versus polyethylene glycol, you can determine how nitrite would partition in these two materials. With alginate, it basically partitions just like it would with water. You see no difference. The concentration of nitrite within the encapsulant is the same as the concentration outside the encapsulant. With polyethylene glycol, it's a little different. It's a little lower. And so if you had a system where nitrite was, say, being consumed more slowly, it was potentially inhibitory, you may want to think about using an encapsulant where it actually can help mitigate that by excluding the nitrite. When we look at wastewater organics, you see something different, but more dramatic, where the wastewater organics, again, are around 1. We've got a much higher variability here with these experiments, but you've got about the same concentration of wastewater organics inside the bead as outside. But when you have this silica gel coating, it actually acts as this diffusion barrier, and it keeps this material, the wastewater organics, from partitioning into the alginate. And so it kind of slows down that delivery of wastewater organics to the inside of that bead. And what you might want to use this for is if you had a system where you were encapsulating autotrophs and you wanted to lower that wastewater organic concentration, silica gel coating might be a good choice. Or if you had a system like these high strength wastewaters that we deal with, dairy, something like that, it doesn't really matter because the concentration is going to be so high that the organisms are going to be able to consume that material regardless. If we look at some other data, here I'm showing some potential inhibitors, ammonium, copper, and dichromate. We've also done some work with inhibitors. And what you can see is that when we've got our silica gel coating, you see that ammonium in particular is enriched in that encapsulant. It's also enriched in just an alginate encapsulant. And so this is an interaction between the carboxyl groups within that alginate matrix. And so you actually can get more ammonium pulled into that bead than is outside in the bulk liquid. When you look at copper, it looks like it's heavily enriched in the encapsulant, but when we did some additional experiments, we found that it was actually bound in the encapsulant. So what's happening with the copper is it's actually sequestering that copper away from the organisms, and it's not bioavailable. So essentially it protects them. When we look at the dichromate, it's similar to what we saw with the nitrite in PEG. It's repelled from the encapsulant because of the negative charge. And this, again, can help mitigate dichromate inhibition within one of these beads if you've got the silica gel coating on the alginate. This is some data that we did on based on that work to look at how these different encapsulants work in terms of mitigating inhibition. And every one of these points is a different experiment. We're looking at the normalized rate of hydrogen production. So these are anaerobic hydrogen producing bacteria. And what you've got on the y-axis is it's a log scale, and it shows this normalized hydrogen production rate, so essentially activity. And then along the x-axis, we've got the concentration of these various inhibitors. The black squares are our encapsulated bacteria, and the red circles are suspended bacteria, so the control. And what you see is with the copper, again, this copper is being sequestered, it's being tied up within this encapsulant. And so we can add really inhibitory levels of copper. And in the encapsulant system, the bacteria are not at all impacted. And so they're very effectively protected against this copper inhibition, where you see with the suspended growth system, these bacteria are really highly impacted by this copper. With the dichromate, you see the same pattern, but it's not quite as dramatic, which makes sense based on the numbers I showed you. It was a much smaller difference. Here again, your encapsulated bacteria are protected, not to the same extent as they are with copper. With ammonia, if you remember that ammonia was actually enriched in the encapsulant, you see the opposite trend. And here we've got sky-high concentrations of ammonia. We're looking at anaerobic systems and systems that could be like a meat processing wastewater, something like that, that could have very high ammonia. And what you see is that the suspended organisms do much better than the encapsulated organisms. They're running into trouble here because, again, this ammonia is being enriched inside that encapsulant. Whoops, my mouse keeps going crazy here, sorry. And then with the chloroform, we saw that the partitioning coefficient was close to 1. And so we really don't see much difference between the suspended and the encapsulated organisms. We can also, though, take advantage of this information to improve the activity of encapsulated bacteria. And so I told you that we've also done work on aerobic ammonia oxidizers. They're actually at an advantage if you can enrich the ammonia into the encapsulant. So it's delivering higher concentrations of their electron donor. And so here again, I just show that that partitioning coefficient, we've got higher than 1. It's not a lot higher than 1, but you do get enrichment of the ammonia in the encapsulant, and that can be an advantage for growing ammonia oxidizers. So that's all I'm going to say about that. We've done more work and on more different encapsulants, but what I really just want to highlight is the importance of the encapsulant material and how that interacts with the environment around it in order to help us understand how to protect the bacteria, how to enrich their activity. And so we feel like you can use some very simple experiments to really get at how this encapsulant is going to perform. So now I want to talk a little bit about this biomass encapsulant interaction. And with this, again, we're looking at the escape of the biomass, we're looking at migration or differential growth, and we're looking at just growth in general. How do these organisms grow within this encapsulant? And so with these experiments, we're looking at, again, escape, growth, migration, community shifts, and we measure this by looking at organisms that leak over time, looking at measuring copy numbers of a particular organism in the encapsulant over time so we can monitor growth. I've got a postdoc who's done some really neat work, I think, on using fish to visualize encapsulated organisms over time and using that to actually, now he's starting to quantify how different populations differentially grow inside these encapsulants. And then we've sequenced the encapsulated organisms over time as well. So this is some data for from some really early experiments that we did where we were looking at leakage. And with these, again, we're looking at hydrogen utilizing bacteria and looking at the effect of different crosslinkers in an alginate matrix and also this silica gel coating to determine how that impacts leakage. And what we saw was that the biomass was pretty well retained in quiescent systems with these alginate beads. And the different crosslinkers definitely impacted leakage. What we saw was that this, this uncoated calcium retained the bacteria fairly well. The uncoated strontium, we leaked more biomass, and the uncoated barium cross-linked alginate, we leaked even more biomass. This is, again, over 30 days. And so as long as the biomass is able to grow, it should even out. And something like the calcium system should work out okay. What we also saw was that when we added this silica gel coating, for the most part, it effectively retained the biomass further. And so when we coated these calcium cross-linked or strontium cross-linked beads, we were able to really effectively retain that biomass. It essentially didn't leak at all. With the coated barium, that silica gel didn't last well on that barium cross-linked material. You can get swelling sometimes, and then that silica gel is actually fairly stiff and sort of pops open like a pea shell. And so there's some interaction between the inner encapsulant material and that coating. But we were able to see kind of how this interaction worked in terms of the tightness of this cross-linking and then also the effect of this coating. What we didn't know was this was small scale lab systems. And what we didn't know is whether this would work as well in an actual system. And so again, going back to that brewery system, we ran experiments with digester sludge that have been encapsulated, and we subjected that to actual high strength brewery wastewater that was coming in extremely variable over time. ran the experiment for about 160 days. And we took the beads out over time, and we quantified the methanogen 16S rRNA gene copies in those encapsulants. And what we saw was that we had clear leakage of the methanogens from those beads, that the gene copies decreased about an order of magnitude over the course of the experiment. But in addition, and probably more importantly than this leakage that we saw from individual beads, was what you see here. We saw a lot of breaking of the beads. And so you can see kind of little chips of these beads in here. And these beads are breaking over time because of the mixing in this reactor. And so we had both breakage and leakage from these alginate encapsulants, telling us that this really, in the end, isn't going to be an encapsulant that's going to get us where we want to go in terms of performance. So based on that, we've been testing alginate-PVA blends. We've also been testing PEG, and we've been testing some unique kind of core shell systems. I'm just going to show a little bit of data on the alginate-PVA blends. And what we've been doing here is looking at different types of cross-linking conditions, different biomass concentrations, so that we can achieve these goals of stability and strength over time. This just shows some pictures, and you can see when we cross-link for about an hour at a pH of 6, you have pretty mushy beads after about 4 weeks, and these have pretty much completely disintegrated into like a jelly. When you cross-link for 1 hour at a pH of 4, it's pretty inhibitory to the microorganisms, and the beads are a little bit better off, but not as strong as we need them to be. You can see, particularly with the low biomass system, these beads swell a lot, and then they end up leaking. When we cross-linked overnight at a pH of 6, you end up with much stronger beads. You can see these are all pretty much intact. And we're able to retain that biomass effectively over those 4 weeks. Encapsulation chemistry, I mentioned the cross-linking and polymerization. With the PVA, you cross-link with boric acid, which can be inhibitory, and with the PEG, you polymerize with an activator that is a strong oxidant, and that can also be inhibitory. And so it's this sort of balance between a bead that'll last and then something that won't kill your bacteria that you're trying to encapsulate. And so we've done a lot of experiments like this where we've looked at batch systems when we encapsulate using different chemistries, and these are all different chemistries. The blue one is our alginate control, which is a kind of a friendly chemistry. How inhibitory is this over time? And again, these are our hydrogen producers, and we measure this just by looking at the hydrogen produced over time. And then in some of these systems, we refeed them to see if they recover slowly. And so what we were able to see is that with this low biomass, but overnight pH 6 cross-linking, where we saw that excellent stability, we also see that pH adjustment really protects the organisms from too much inhibition. You get some inhibition early on, but they're able to recover and come back. And so we kind of pair these two types of experiments to figure out how we can encapsulate organisms in a more biologically friendly way that will also provide strong encapsulation chemistry. And so from this, we see that we should be able to create an encapsulant that is non-inhibitory enough, strong enough, and retains the organisms. But this is an area that we're still working on. And so what we really want to know is that enough? If you can encapsulate certain organisms, will they then grow and adapt once they're encapsulated? If we can get some seed culture in there, are we good enough to handle different variable influence? And so one of the first questions is, how do the organisms grow in these systems, and do they grow in a predictable way? I'll get to the predictable in a minute, but looking at how they grow, my postdoc Philip ran some experiments where he encapsulated some pure culture AOB, ammonia oxidizing bacteria, and ran some batch experiments, and he saw that the ammonia went away, the nitrite formed as predicted. And then when he took these beads over time, he would take individual beads out and he would extract the DNA and measure the ammonium monooxygenase copy numbers. He could see that there was growth over time. And so these organisms are growing within this encapsulant bead. And so that is, that's a positive sign that they will in fact grow, even if they leak or some of them die off during that encapsulation process. He also did some really nice work. I hope that this image comes through. It's always tricky to see these, I think, online. But he also did some really nice work with fish, where he would slice these beads in 10 micron thick slices, so thin slices, and he could look at them over time. And with this, again, we're looking at the pure culture of AOB. Now he's looking at multicultures. But what you can see is at day 0, hopefully you can see this, the bacteria are pretty well distributed throughout this, this slice of bead. And then as time goes on, this is at day 3, you can see that the signal is much brighter on the outside of that bead, where these AOB are in closer contact with the oxygen. That oxygen is diffusing into that bead, and you can see that it leads to differential growth of those AOB on the outside of that bead. And so that again tells us that we can go back to our encapsulant chemistry and look at this diffusivity data and hopefully predict where we'll get certain zones for certain types of organisms. We also know that they can change, but we wanted to look and see how they adapted, how those microbial communities adapted with time. And so for this experiment, what we were doing is taking this big messy experiment, and we had our reactor containing the encapsulated biomass, and we would, we would take samples of beads out of this reactor with time, and we would sequence those beads. And then we would also take samples of the biomass in the same encapsulation reactor of the organisms that were free flowing, that were growing outside of that encapsulant. And we sequenced those organisms as well. And then we also had this parallel anaerobic membrane bioreactor, and we could then take samples of the biomass within that reactor with time and sequence those organisms as well. And so again, we sampled both the beads and outside of the beads in our encapsulated biomass reactor. And then we also sampled the suspended growth in the ANMBR, and we performed deep sequencing on this, just looking at the 16S rRNA gene to determine how these communities adapted with time. And so this is a Brake-Curtis beta diversity plot, and I'm going to talk you through this. This is how the community structure evolved with time. So this would be an early sample, and then with time, the community evolved and changed. And you see with these communities plotted over here, they're growing and changing with time in this direction. If we look at the orange biomass, this is the encapsulated anaerobic community. So the DNA that we extracted from those beads with time. And you can see that they adapt in this particular way where they're changing with time. So we can see that those communities do change with time. But if you look at the green dots, which are the suspended anaerobic community outside of that encapsulant, it adapts in a very different way. The beginning community is quite different. This likely comes from the wastewater itself, and it changes in a very different way than the encapsulated community does. And then if we look at the purple squares, this is our parallel ANMBR anaerobic community. So it adapts in pretty much the exact same way as our suspended growth in that same reactor, But in our ANMBR, we have a very high biomass concentration. And in our encapsulant, we have a very high biomass concentration. And then the biomass concentration suspended outside that encapsulant is pretty low. There's not a lot of biomass there. And what we saw, which was not particularly positive, was that actually the adaptation that we saw in this ANMBR corresponded to really excellent performance. We saw really good COD removal, we saw really good methane production, we saw a very stable performance in these communities. And when you look at the performance in the encapsulation system, we really had pretty poor COD removal, probably closer to 50 or 60% COD removal, whereas we had over 90% in the ANMBR, and we had much less methane production. And so what this tells us is that the initial community composition seems to be really critical for optimized performance. The biomass can grow, and it can grow differentially in that bead, and it can adapt within the encapsulant, and all of that's really exciting. But if you seed the reactor with the wrong community, it's not going to adapt in a way that will facilitate good performance. And so that's also something that we're working on now, is trying to figure out how do you tailor an encapsulation, a community to be encapsulated, so that you ensure that you have robust performance, but also flexible performance. And so now I want to kind of end here with this idea of predictable performance, because all of this is interesting. It's getting us closer to getting a system that'll work, but it's all very specific to a particular environment that you're looking at. What my student Philip, or my postdoc Philip's done, is he's adapted a 1D biofilm model in Aquasim. And what he's done is, this is work that we did with Rob Nuremberg at Notre Dame. We actually sort of created this alginate biofilm. And the alginate biofilm contained biomass and contained alginate particles. And that allowed us to account for the absorption of ammonium into this alginate component. It also allowed us to have room for the organisms to grow and move. And it could then also account for the different diffusivity of different species in this alginate biofilm compared to a traditional biofilm. And we use this to predict activity and growth within the system. And so I showed you this plot before, and this is a plot of ammonia going away as measured, and the nitrite being produced, and then the biomass growing with time. And so these are from a very simple single culture system, looking at a pure nitrosomonas culture. And what we saw was, this is the model prediction, and this isn't fit based on the data. This is plugging in coefficients from the literature for nitrosomonas. And what you can see is here, with this very simple batch system, the model predicts performance really well. And I think what's more interesting is if you look over here at the growth within the encapsulant, what you can see is that the model, these lines, predict differential growth. And so 1 millimeter is at the shell, the outside of that encapsulant bead. 0 millimeters is at the very core of that bead. And so what you can see is that the growth in the core is predicted to be less, and the growth in the shell is predicted to be greater in the end after 5 days. And our data was a pretty good match to the 0.5 millimeter data, which is kind of what one might think of as an average, which makes sense because here we were extracting the DNA from the entire bead, so an average. So what's cool about this model is that it really lets us kind of see this differential growth. And then when you look at the modeling results, it predicts this DO differential across the bead. So I showed you the differential growth that's predicted, but you also see that there's high DO on the outside of the bead. That's up here at 1, and areas of low DO inside the bead. And this is over time. So as the activity is increasing, as the organisms are growing and degrading that ammonia, you get this low spot of DO inside the bead. And then as the organisms slow down, they stop consuming the ammonia that's provided, then that DO can kind of rebound and go throughout the bead again. And you see a simultaneous change in the pH throughout the bead. And these results were qualitatively verified with this fish data I showed you before. We could see this sort of differential growth happening. And then we also performed microsensor measurements on an alginate bead that had biomass in it and an abiotic control bead. And what you could see is, again, here at 0, that's the outside of the bead. And here at about 1 millimeter is the bead core. And so you can see that we see the DO is high, it drops in that boundary layer around the bead, and then it dips to a low point at the center of the bead, and then it comes back up. And with the abiotic bead, where you don't have any biological activity happening, that DO is pretty constant throughout that bead. And so this is definitely qualitative, but it gives us this idea that this model is doing a pretty good job. It looks like it's doing a really nice job with prediction of growth in terms of the copy numbers, but then we also see this differential growth, which is pretty neat. Philip has expanded this model to include both AOB and Animox now, and he used it to look at performance in a flow-through reactor. And with this one, I naively thought that if we had low DO outside of in the bulk solution, that we would get this AOB activity on the outside of the beads, Animox activity on the inside of the beads, and we'd get really good total nitrogen removal. And that was not what we saw. When we saw no aeration, we saw really no ammonia degradation at all. This is the model prediction. When we saw low aeration, we saw, again, really no ammonia degradation at all. And well, we saw a little bit, and then we saw, I guess, some nitrite production, but really no Animox activity. And then when we saw high aeration, again, we see this decline in ammonia and an increase in nitrite, but the Animox aren't really doing anything with this. And so this was actually what he saw in an actual flow-through reactor, pretty similar patterns. It didn't work. And so what's nice about the model is that he's built in inhibition as well into the model. And so what you can do is, by looking at the model, you can kind of figure out what went wrong. And what's happening is you need to have this initial ammonia degradation, but you can't let the nitrite get too high, or else it starts to inhibit the organisms. And so he used this model then to identify what might be a better option. And so we started going back to a batch type system with initially high DO, and he could then kind of tweak what sort of biomass levels you might want in this bead. High AOB versus low Animox, or low AOB versus high Animox. And what the model predicted was that if we have initially high DO in this batch system, the DO drops, and you get this in a high AOB, low Animox system, you get degradation of the ammonia, you get production of nitrite, and that nitrite, it sticks around. It doesn't really degrade very effectively. With low AOB and high Animox, we're able to knock down that ammonia quickly, develop a little bit or produce a little bit of nitrite. And then at this point, those Animox organisms can start to degrade the nitrite and the ammonia and keep the total nitrogen level very low. And so we use these model predictions then to run experiments to see if this was accurate. And it seemed pretty good. We've got here, you can see these are the batch results of these encapsulated, these co-encapsulated organisms where we have high AOB, low anammox, low AOB, high anammox. And what you can see is here you see that ammonia go away and we get that nitrite production. Whereas in this system with that high anammox, you see the ammonia drop and then a little bit of that nitrite form. And then that nitrite is really kept low pretty much the whole time. And we see that additional ammonia degradation as anammox bacteria are consuming that nitrite. And so this suggests you're not going to run a large wastewater treatment plant in batch mode, but it does suggest that pulse DO could have the same impact. And contrary to what I was initially thinking, we really need that high initial DO to kind of jumpstart the aerobic ammonia oxidation process, so then the anammox bacteria can take over. So I'm getting close to the end here. What are the next steps? We're doing further modeling and experiments to optimize this BNR process with the co-encapsulation. And now Philip's doing slices throughout the bead so that he can look at differential growth of the anammox and the AOB using different fish probes with depth. We're doing additional development and testing of new robust encapsulants. We're doing a lot of these core shell systems and then trying to refine the PVA-alginate mixtures. And then optimizing this encapsulated community so that it can better adapt and we can get good performance instead of this kind of adaptation in the wrong direction in terms of performance. And with that, we're really looking at kind of creating communities and then seeing how they develop with time with different outside chemistries. And so in the end, the goal that we have is this predictable small footprint, low energy BNR and industrial wastewater treatment. And that's it. I'll acknowledge my collaborators. Bill Arnold has been along for this whole journey, been a great collaborator. And Satoshi Ishii at U of M has been a great collaborator on the nitrogen removal work. Rob Nuremberg and his postdoc, Patricia at Notre Dame, were really helpful with the modeling work. And then I have my army of graduate students and postdocs that have been working on this. Kwong graduated a while ago and did the early work on this. Philip's been working a lot on the BNR work. Simeng and Tony have worked on the brewery and the community work. And then I've got Gretchen now is working on my encapsulant chemistry, and Olivia has been working as a great undergrad in the lab and had funding from lots of different sources, most recently Department of Energy. So with that, I'm happy to take any questions.